Okay, here we are with what will be the third and final part of the photosynthesis online lecture, chapter 10. So at the end of the second part, we had basically concluded talking about the light reactions. We had basically two energy products from the light reactions. We had ATP and we had NADPH. And remember, ATP is going to supply the energy to run the next step of photosynthesis, which is the Calvin cycle. It's anabolic, endergonic, it requires energy. And NADPH provides electrons and hydrogen as building material. Remember, CO2 comes into the Calvin cycle, but that only gives you two elements, carbon and oxygen, that you need to make a sugar. You need a third one, hydrogen, and that's what NADPH provides, along with high energy electrons that get loaded into the sugar molecule as the energy content of that food. Okay, so the Calvin cycle, like the citric acid cycle, is going to regenerate its starting material after molecules enter and leave the cycle. So it's a cycle. It starts with a gateway molecule, and it's going to convert that gateway molecule to various intermediates as the cycle does its thing, and the back half of the cycle will rebuild that gateway molecule. Now, if you remember, in cell respiration, the citric acid cycle, the gateway molecule was called oxaloacetate, right? We talked about that. Okay, here in the Calvin cycle, we have a different one. We have a molecule called RUBP. Okay, RUBP is our gateway molecule here in the Calvin cycle. All right, so basically we're going to have carbon dioxide attached to RUBP, and that is the fixation step. So RUBP is important for carbon fixation because it is what accepts the carbon in inorganic form as CO2, thereby converting it to organic form. Okay? All right. So basically what's going to happen here is that carbon enters the cycle as CO2, and that's inorganic, and ultimately it's going to leave the cycle as a 3-carbon sugar called G3P. That sounds familiar. It's because we've already seen it. If you remember the first half of glycolysis, the investment phase, at the end of it, we had 2 molecules of G3P. They then dropped into the payoff phase, and all the rest of that magic stuff happened. Okay, well, here we see the same thing. We see a molecule of G3P. This, again, is just reiterating some of the similarities and the linkages between photosynthesis and cell respiration. All right, so the Calvin cycle has 3 main parts. First one is the carbon fixation step, like I said. Okay, that's where inorganic carbon, the CO2, becomes organic. Second, we have the reduction phase or reduction step. Okay, that's where the NADPH comes in and dumps off electrons. All right, and third, we have the regeneration of RUBP. That's our CO2 acceptor. Okay, so this is the Calvin cycle in a nutshell. All right, so just to remind you now, here we are in the Calvin cycle. Okay, so we have our ATP and NADPH coming in from the light reactions. We had CO2 coming in from the atmosphere. The leaf absorbed that. What we get out is 1 sugar. Okay, all right. So again, the Calvin cycle is a little complicated. It's not actually as complicated as, I don't think, as the citric acid cycle and cell respiration. But in this next figure, I'm going to show you what's important. Okay, so focus on the things I tell you to know the names I tell you to know. You don't have to know about structures. In fact, this next image doesn't even show you structures. Focus on the overall flow and pattern of things, all right? Okay, so this is the Calvin cycle. So we have our CO2 coming in. Okay, that's our source of inorganic carbon. And we have 3 of them. All right, why? Because our output is going to be 1 G3P sugar. So we see 1, 2, 3 carbons there. That means we need 3 CO2s to supply those 3 carbons. Okay, now over here on the left, we have our RUBP molecule. Notice there are 3 copies of it, and it has 1, 2, 3, 4, 5 carbons. So 3 copies times 5 carbons each. That's 15 carbons, all right? Okay. To each one of these RUBPs, a single CO2 is added, and so that gives you this molecule here. Okay, it's an intermediate, it's highly unstable, doesn't last very long. Don't worry about the name, it's not even shown. But now we have 3 6-carbon compounds. Now we have 18 carbons. That shows you we did add 3 more carbons to the 15 we had here. Now we have 18 in these 3 intermediates. But like I said, that intermediate is very unstable, and it essentially snaps in 2 very quickly, and now you get 6 of these 3-carbon intermediates. 6 times 3 is still 18. We still have our original 18 carbons, right? Okay, ATP from the light reactions comes in, 6 of them, and donates a phosphate to each of the 6 of these. That gives us these intermediates, or this intermediate here, 6 copies of it. Again, don't worry about the name, not important, okay? All right, but notice it has 2 high energy phosphates, each one of them does, so it's very energy rich. All right, this is our reduction phase. NADPH comes in, dumps off electrons, gets oxidized to NADP+, and at the end of that, we get 6 G3Ps, because a phosphate comes off of each one of these, leaving this, 1 phosphate. That's G3P. So again, 6 times 3 carbons, 18. One of those comes out of the cycle and will be used to build glucose and other organic compounds. Okay, oops, sorry. So remember, glucose is shown in the reaction as the final product of photosynthesis. Technically it's not, though. G3P is, but that G3P mostly gets built into glucose. That's why we show glucose in the reaction as the final product. But that G3P then is the building material to make lots of other things besides glucose. Other organic compounds, proteins, other complex sugars, fat molecules, nucleic acids. The organic carbon structure of all of our molecules is possible because of that G3P sugar that comes out of the Calvin cycle. Okay? All right. Now we're over here to phase 3, regeneration of our CO2 acceptor, the gateway molecule, RUBP. So we now have 5 of our 6 G3Ps left, so 5 times 3 is 15 carbons. All of the rest of this is really just sort of a rearrangement, okay? So we have 3 ATPs coming in, dumping off phosphates, and that builds these 5 3-carbon compounds into 3 5-carbon compounds. 5 times 3 is 15 carbons, 3 times 5, 15 carbons. So same number of carbons, we've just built 5 3-carbon compounds, small compounds, into 3 larger 5-carbon compounds. 3 copies of RUBP. Now we're ready for the next run of the cycle. 3 more CO2s drop in, okay? All right, but the important thing now is this 1 G3P sugar that's come out. All right, so I have a Bioflix animation here to show you next, okay? And this is going to summarize basically everything we've talked about. It does a really good job of showing you the light reactions, electron transport chains, all that. Kind of glosses over the Calvin cycle again because that's kind of hard to show in one of these animations. So let me play this and then we'll go on and talk about a few other things, and that'll be it. All right.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The 2 green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the 2 photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
Okay, so one question that students often ask is, why does the plant need a Calvin cycle? So the Calvin cycle produces a sugar molecule, and like it said, the video said, that G3P can then be used in the mitochondria of the plant to produce ATP. Well, if photosynthesis, the light reactions, are already making ATP for the plant, why does it then need to go on to make a sugar molecule which then goes into the mitochondria just to make more ATP? That seems kind of redundant. The Calvin cycle is important because it provides carbon skeletons. It is the only thing that provides organic carbon skeletons that can be used to build sugars, proteins, fats, and nucleic acids. So it's a building process in addition to a process that provides the plant cell with ATP. Okay, that's the real key importance of it. Without the Calvin cycle, you couldn't build the bodies of animals because you would not have proteins, sugars, fats, nucleic acids with which to build. So we wouldn't exist. Okay, so last thing to talk about is just sort of a review then of photosynthesis. So the energy entering chloroplast as sunlight gets stored as chemical energy in organic compounds. Sugar made in the chloroplast supplies chemical energy and carbon skeletons to synthesize or make organic molecules of cells. And plants store excess sugar as starch in structures such as roots, tubers, seeds, and fruits. That provides food for lots of different things. And in addition to food production, photosynthesis produces the oxygen in our atmosphere that we then use in our mitochondria to break down the food, again, that plants give for us. So you see the plants are absolutely essential to the complex life on this planet. We can do without, or plants can do without us, but we cannot do without plants. Okay, that is the end then of the third part of the photosynthesis chapter. I'll be working on the next one, which is chapter 12 on mitosis, probably early next week.